Bueno, bienvenidos a tu nuevo ciclo de charlas e ideas. Gracias por la paciencia. Nos pasamos un poquito del comienzo de la charla, les pedimos disculpas, pero faltaba nuestra experta en YouTube, así que hicimos lo que pudimos. Ahora yo voy a pasarla en inglés, pero primero para presentarlos un poco, va a dar una charla el doctor Wang, que está trabajando ahora en colaboración con el laboratorio del GIS, que es grupo de Ichnología sedimentológica, y especialmente con la doctora Carolina Gutiérrez, que después si quiere nos cuenta lo que estuvo haciendo en Brasil, sí, que se vinieron del Congreso de Ichnología. Y ahora paso para presentar al doctor. Dr. Zekun Wang obtained his PhD from the College of Engineering at Peking University, China, where he later worked as a research associate. Currently, he is an international fellow of the Royal Society and works at the Natural History Museum in London. He proposed multiple computational algorithms for modeling particle flows, which are now used to study the formation of fossil traces dating back to the Ediacaran period and Cambrian period. He also innovatively employed various mathematical approaches, including frequency analysis, correlation analysis, and vector analysis in quantifying fossil trace morphologies, through which he established a link between the biological signatures of trace makers and patterns existing in locomotion traces. These results help calibrate the evolution of behavior, anatomy, and locomotion of early eumetazoans and establish a quantitative framework for the classification of fossil traces. This interdisciplinary work helped him win the Excellence in Research Award from the International Association of Applied Mechanics, the Emerald Literary Award, and the MSCA Fellowship. So we're gonna have some time after the talk for questions, and we invite everyone watching through YouTube to leave their questions through the chat. So now we welcome Dr. Zekun Wang.
Thank you, everybody. Thank you. Yeah, I'm Zekun Wang from the Natural History Museum in London. Yeah, and I work at the interdisciplinary science between computational techniques and paleontology, especially for the trace fossils. Okay, today I will talk about my recent research in quantitative technology, which is the discipline that studies the trace fossil with numerical and analytical methods. Okay, yeah, when we go to a natural history museum, you can always see different kind of fossils, from the big dinosaur skeletons. Is it a thing? To the earliest birds or the earliest flowers. And these body fossils inform us of the major evolutionary events that happened during Earth's history, from the massive radiational diversification, like Cambrian explosion, to the massive extinction events, for example, the extinction event that happened at the Permian-Triassic boundary. But there is another kind of fossil that is also quite valuable for paleontologists, that's trace fossil, which are the fossilized interactions between the organism and their environment, including food and substrate, especially for the locomotory trace fossils, like the trackways left by dinosaurs, or arthropods, or mammals, or even invertebrates, other invertebrates, or the burrows, or the trails, even their eggs, their poops, their nests. They're all fossilized interactions between these organisms and their environment. Therefore, they can provide unique insights into the evolution of their, for example, their behavior or their locomotion during the Earth's history. Yeah, and these trace fossils also appeared very early. They can be dated back to Ichnology, and they flourished during early Cambrian with all different kinds of morphologies. And some of the trace fossils even appeared much earlier than the primitive trace makers, like the locomotion trace of trilobites, Louisiana, or the resting trace of the trilobites. They appeared earlier than their body fossils of the trilobites. And by calibrating the diversification, we can have a better understanding of the evolution of behaviors or tearings or niches or mode of these animals. But there is one challenge in the study of trace fossil, that is, there is no one-to-one mapping between the trace maker and the trace fossil. That is, for example, a charla, it can create different kinds of traces on different behavior or on different substrates. But animals from absolutely different phyla, like these sand fleas or the mud snails, they can create similar traces, judging by our naked eyes. Therefore, our diagnostics for the trace fossils tend to be subjective, and it can vary from one person to another. Several typical classifications include, behavior classification, like the pure locomotion, genia, or the grazing, or as genia or other behaviors, escaping different behaviors. We also classify them with the architectural design, that's the morphology, like it's a U-shaped burrow, or a straight burrow, or a trail or trackways. Yeah. So also classify the trace makers with tearings pattern. For example, like the male phonobiotibators or biodiffusers or conveyors, they are a classification of the tearings in these trace makers. But these diagnostics are basically made on our observation and assumption of these trace fossils. So they can be, to some extent, subjective. So more and more people are now seeking quantitative methods to describe and diagnostic and also interpret these trace fossils, especially for those simple horizontal trace fossils that seems to lack information. So today, in my talk, I will introduce 3 different approaches that can help us quantitatively understand the paleobiological significances of these trace fossils. This includes the frequency and occlusion analysis, the trajectory smoothness of the trace fossil, and some numerical approaches that can help us reconstruct the formation process of trace fossils. Okay, first of all, let's talk about the frequency and occlusion analysis. This is a signal, no matter what kind of signal it is. It's an audio signal, a sound, or the image, or even the stock signal or the velocity of friendship, no matter what kind of signal it is. As you can see, it has 2 different scales of self-repeating patterns. This is the large scale self-repeating patterns. The period is around 2, and this is a small scale repeating patterns, and the period is around 24. But if this signal becomes much more complicated, if it would be difficult for us to recognize the underlying self-repeating patterns of this signal, it's complicated. Oh, I forgot to say that this signal, it evolves with time or distance. So we call it a signal in the spatial or temporal domain. Therefore, we use a mathematical technique. We call it Fourier transform to transform this signal from the spatial or temporal domain that increases with distance and time to the frequency domain. So in this case, we can identify the hidden frequency behind the signal. As you can see, there are 2 peaks in the frequency spectrum. The first one is 0.5, which is the inverse of 2, because the period is the inverse of frequency. So 0.5 is 1 divided by 2, the inverse of 2. And another peak is the inverse of 4. And the magnitude of these 2 peaks is the same, because you can see the coefficient. So by performing the Fourier transform, we can actually transfer the signal from a spatial temporal domain to a signal domain that help us better recognize the underlying signature, the underlying self-repeating patterns hidden behind a signal. So what is the signal that we can use for analysis in trace fossils? Could it be the coordinates? No, because when we do a Fourier transform, the signal has to be bounded, has to be limited within a specific range. And the coordinates, theoretically speaking, it can be infinitely large. So we use the turning angle signals, because the turning angle is bounded by positive and negative 180 degrees. It's abundant signal. Yeah, that we can use Fourier transform. Therefore, we need to calculate the turning angle series. For example, this is a trace left by a modern gastropod. And first of all, we need to normalize this trace because sometimes the animal is a little bit bigger and sometimes it's a little bit small. But for the same animal, we assume that they have similarities, like in terms of the body size or the locomotor mode. They have similarities. So we need to normalize the trace. How do I do this? This is L is the real length and W is the trail width. So the normalized distance is the real distance divided by the width. So if the normalized length is 2, it means the length is 2 times the width. Then we can have these sampling points. We can calculate the angle between two sampling points using unit vectors and trigonometric functions. And therefore, we can obtain a turning angle series step by step. This turning angle series should obey the right-hand rule. This means if it turns clockwise, that if they turn anti-clockwise, we grab price to y anti-clockwise. So this points to therefore, if it turns anti-clockwise, then it's a positive value. If it turns clockwise, then it's a negative. In any case, we can obtain a signal which is bounded by positive and negative 180 degrees. Then we can use Fourier transform to transform this signal and see what happens. Okay, this is the formula. It doesn't matter. Actually, many packages like R packages or MATLAB, they have some functions. You can use it directly. So you can ignore the expression. But in any case, yeah. For example, here are the traces left by 4 typical of modern animals. This is the trace left by an isotope, which is a small. As you can see, there is no major frequencies in this frequency spectrum. This means there is no self-repeating patterns in its turning angle series. No self-repeating patterns in its trajectory. But for nematode, as you can see, there is only one major peak in its frequency spectrum, which is around 0.75. And we can calculate the period, dimensionless period, which is the normalized period, which is the inverse of the frequency, and it's around 13. What does it mean? It means the length of a 4 meander, the length of a 4 wriggling is around 13 times of the body width. And for qualities, the looping qualities, they tend to loop, reduce to more Texas. This value is much smaller, but for gastropods, it's different. As you can see, there are 4 to 5 different major peaks with decreasing amplitude in the frequency spectrum. This means the pattern is actually a fractal pattern with 4 to 5 major different self-repeating patterns at different scales, at different orders. So it is a fractal pattern. It is a space filling curve. This is related to their kinesis. Kinesis is a logical term. It means making frequent terms or frequently changing directions in response to a changing density of stimuli, for example, food resources. This is very typical of mollusks, different mollusks, gastropods or the slugs, because they graze on the substrate. Okay, another approach is the autocorrelation function, autocorrelation analysis, which is calculated by this formula. This is a very interesting formula. You can take a close look at it. It tells you the correlation between 2 points along a signal. So this delta L is the distance, the normalized distance between 2 points in a signal. If it equals to 0, this means 2 points is overlapped. When it is 0, you can see the denominator is the denominator. So this coloration is 1. It means this function reaches maximum coloration when the 2 points are overlapped. If 2 points is close to each other, they tend to be in the same structure. They tend to have the same turning direction. So after taking an average, this value should be, after normalization, should be between 0 and 1. If 2 points are far away from each other, they can either have the same turning direction or different turning direction statistically with the same probability. So after taking an average, so this value should be around 0, oscillating around 0. So as you can see, they can all decrease to 0 when the distance between two signal to 2 point is long. So for isopod, as you can see, this coloration function decreases rapidly. It means it decolorates. The 2 points are not related to each other, even in a very short distance. So it decolorates very fast. And we can define a coloration scale at its first minimum value, which is 1.4. This means the typical scale of a term that by isopod is around 1.4 times its body weight. Sharp. But for nematode, as you can see, this minimum scale is around 6.4, which is exactly half of the normalized period we identified with the frequency spectrum. Why this? Because in an intact meander, here it's anti-clockwise, positive, but here it's clockwise, it's negative. So the signal decolorates after it enters the path. That's the reason why this coloration is one half of the normalized period, which we calculated just now. And for polycase, this is larger, and for gastropod, it's around 4. This tells you that the flexibility of gastropod is between the isopod and nematode. Therefore, we can use these two approaches to examine some trace fossil record from early Paleozoic. These are, for example, 4 typical Gordia. This Gordia nodosa from Cambrian, as you can see, it only has it shows resemblances with the traces that apply polycase, looping polycase, and this one, Gordia. It shows sharp terms. It shows sharp terms, and there is no major frequencies in its frequency spectrum. It means there is no self-repeating patterns in its trajectory, and the coloration scale decreases rapidly. Quite similar to a small arthropod, the traces are left by small arthropod. And this Gordia marina from China, it has several different major peaks, telling you this trace maker could also have some kind of a kinesis, grazing on the substrate with a space-filling curve, with a fractal curve. And it's probably a mollusk, not necessarily a gastropod. Mollusk with a grazing habit, muscular soul. And another Gordia marina is quite similar to a nematode trace. It's from Devonian, so it's possible. And at least it tells you that the anatomy of the organism, they use this region as a propulsion to go forward. So as you can see, these approaches can help you identify the putative trace makers or the anatomy of at least the anatomy. Then we can make comparisons between the earliest appearance of certain trace fossils and their putative trace makers. And more importantly, we can use these two metrics to quantify the classification of trace fossils, locomotive trace fossils. For example, here, the x-axis is the coloration scale. The y-axis is the major frequency. For example, for this Gordia left by probably arthropod with no self-repeating patterns, its coloration scale is around 1.4, and it doesn't have major frequency, so it spreads from 0 to 1. And for this Gordia marina, probably left by mollusk, then its scale is the coloration scale around 4, and it has 4 major peaks. So in any case, each trace fossil, they If they are correctly diagnosed, then it should occupy a unique region in the space map. Therefore, these matrices actually help us quantify the classification of simple traces. Oh, I actually, it's published and it's I made it into a software, so you don't need to do the mathematics. You can just click to calculate. You can download the free software link. Okay, let's go to the second part. It's smoothness. First of all, I need to introduce two different auxiliaries. Some Ichnologists use smoothness, but mathematically speaking, it's roughness or superficial roughness. It tells you either the variance of its boundary or the variance of its relief, its superficial conditions. And mathematically speaking, we call it a superficial roughness. But what I'm going to talk about today is not this roughness. It's the trajectory smoothness. It means whether a line has abrupt turns, whether it's shaky. So as you can see, by descriptive language, it is very hard to tell you what is smooth. So we need some quantitative methods. For example, the first one is a strange definition. Probably didn't see this kind of definition, but it reads very interesting. You can see, we call them C1 continuity or the epsilon double definition. This is the smallest term, and this is the term with one abrupt. So as you can read the definition, it's very interesting. For all extremely small random positive value epsilon, no matter how small it is, it's a random value, but small enough. You can always find another small value delta, which is the distance between point B and point A, that enables the turning angle between these 2 points is smaller than this randomly chosen small positive value. Oh, then it's as you can imagine, if it has a disrupted point here, you can never ever find a second point that complies with this definition. You can never find a point. And the turning angle between them is smaller than this randomly small value. But if it is smooth, well, they are close enough, you can always find another point that enables the turning angles smaller than this randomly small. That's the definition. Very interesting definition. Okay, and by this definition, we can classify the trajectories into, for example, totally smooth. This means each and every point along a trajectory comply with this definition of smooth. Or original smooth, it means part of its trajectory smooth. For example, we can say if there are limited numbers of disrupted points on the trajectory, for example, 10 within 100 normalized distance, then it's original smooth. However, if there is unlimited number of disrupted points, even each and every point is disrupted points, then it's non-smooth traces. Okay, another method is to use the minimal radius of curvature. The radius of curvature is the radius of the circle that attaches itself closely to the curvature. So along the traces, among all the sampling points, you can always find a minimum value of the radius of curvatures. It tells you something about the abrupt terms. And the third method, well, this is a little bit difficult to understand, so I give two examples. It's spectral density, and as you can see, this is no matter it is sinusoidal or radius or something like that, no matter how you call it. It shows sigmoid axis, a guided motion, and it's a smooth trace. As you can see, it has several peaks at low frequency components. Low frequency components means large scales of repeating patterns because the scale period is the inverse frequency. But as you can see, it basically doesn't have this blue line. It basically doesn't have any high frequency components. This means it basically doesn't have small scale oscillations. Smooth. But for this sinusoidognitis, it's basically, you can see now it's shaky, it's unpredictable. It has many large frequency components. And large frequency components means small scale oscillations. It's not smooth. Therefore, we can integrate this curve from 0.5 to 1 to calculate the total energy consumption for small scale non-smooth locomotion. Okay, here are the three approaches. Then we can use these to examine the fossil records from Ichnology to Cambrian. As you can see, here are some results. Basically, all the traces from Ichnology are non-smooth traces with small minimal radius curvatures. This indicates that the trace maker could have a round body without any stereo axis. So around the body, they have some high flexibility. They can terms and these animals, they could be some macro scale single cell organisms like foraminifera or very primitive bilaterals. Not until around 543 million years ago, those original traces with smooth parts but also disrupted points occur. First, statistically, they first appear. And the smooth part indicates the existence of anterior posterior axis that help the organism maintain its trajectory. And also these organisms should have coordinating muscular systems that help them also maintain course. And also simultaneously, those totally smooth traces also first appear. And these traces indicate the existence of biomechanical constraints because, for example, it has a slender body, rigid, elastic slender body. So the mechanical properties prevent it from making abrupt turns. For example, you can imagine for a nematode, it can never ever make an abrupt turn when it's still alive. We have to notice that abrupt turn does not necessarily mean sharp turns. They are different. They can be a small turn, but a discontinuity series. So due to the biomechanical constraints, they cannot make abrupt turns. For example, a typical locomotor mode could be wriggling or peristalsis, and for original smooth traces, a typical locomotion could be gliding, and for totally non-smooth traces, they could be creeping, crawling, like a random walk. Okay, then let's see the results for the total energy for non-smooth locomotion. We just mentioned that this is related to the scale of the locomotion, but what I'm going to say now is also related to the perceptual range of the organism. I did some numerical simulations. Here is a normalized perceptual range. It's a perceptual range divided by the width, the body width. So as you can see, when the normalized perceptual range is very small, the traces is shaky, it's random, it's unpredictable, is not smooth. This is because when you have a limited perceptual range, you can only obtain the information from the stimuli discontinuously and discretely. So you make perceptual decisions once again, compare, trace randomly. But if you have a long-range sensory capabilities, you can sense something that is far away from you, so you can make gradual and continuous decisions and stick to that decision. I want to go there, so I forward to that direction. So this makes the trace a little bit smooth. So this, as you can see, the smoothness of the trace also for the same locomotor mode. It's also a reflection of perceptual range. And as you can see from the results, basically all the traces from the carrion are quite random, shaky traces with high energy consumption for non-smooth locomotion. They are not smooth. Therefore, they say they could have a very limited perceptual range and a very primitive locomotor mode. Actually, this is understandable because for those primitive animals trying to carry in, no matter they are macroscale, single-cell organisms or primitive bilaterals, they don't need to evolve to have advanced navigational capabilities because the map is at least locally uniform. So it doesn't matter which direction they go. They just grade on the map with space filling the curve or just graze the map. So they don't have to evolve to have an advanced navigational capability. But at around 543 million years ago, those regional smooth or totally smooth traces first appeared. This indicates the appearance of long-range sensible capabilities and also coordinated musculatures, hydrostatic systems, and also advanced remote. And the ecology changed. The substrate turned from a probably relatively uniform metaground to a mixed ground. So these animals have to find their food purposely destination. This indicates a relatively early route of chemical information revolution. Okay, you may wonder, how did I get these traces? It's not from fossil record, it's not from observations. Actually, from fossils, we can never vary perceptual range, and for experiments, it is also hard to measure its perceptual range. So actually, I did this with numerical simulation. That is the third part I want to introduce to you. So numerical, experimental, or observational, and serial are three approaches for academic research. And what I want to talk about here now is about the numerical approaches that can be used in trace fossils studies. Well, I have a different background from computational sciences or engineering, say. So before I come to the world of paleontology, I did many different simulations, like the galactic collision with galaxies. And this is a powder-based 3D printing. The powder is gathered behind the nodal, and when the laser hits it, this powder melts into a molten core and forms a molten track, and track by track, layer by layer will form a 3D structure. 3D printing, how it works. And this is a dam break with particles oscillating. And this is a trapetridium. This is going to be published recently. How its structure influences the flows and particle structure, for example, food particles. And this is the formation of the patterns in microbial mat, and this is self-similarity of the weight behind the charla. These are numerical simulations. So it reproduces the similarity in a computer. It's different from experiment or observation or series. It's not like animation, but it produces animation, but it's more accurate. So what about the simulations in trace fossils? The first thing I want to show you is this. This is how I produce those traces. It's the code is written in MATLAB. I can see there is a MATLAB. Some of them use MATLAB. Yeah. So the organism moves towards the gradient of food density. Where there are much food, it just goes to that direction. Due to the heterogeneous nature of the substrate, it can go to a direction. But I can set different body sizes, food densities, collecting rate, or moving speed, and locomotor mode, and perceptual range, etc. The perceptual range is the range that the organism can sense, can collect. He knows where food, and I can calculate the direction with the information within its perceptual range added by a Gaussian distribution. So we can see how these parameters can change the trajectory of the organism. But this is a purely mathematical simulation. What I'm going to introduce to you later is this kind of animation. So probably in some papers and in some films, they also use the computation to make the animations in films. Yeah. The first approach is the coupling between computational fluid dynamics, which solves the motion of fluid, and the discrete element method, which solves the motion of living particles, as well as the organism. The organism in this approach is represented by STL file. This file, you can output this file, no matter from 3D modeling software or CT or laser. Yeah, it's very common file, STL file. And if there is an articulated motion, we can use U-angles to calculate its articular motion. For example, this is a charla feet. And yeah, I will introduce you later. It works on a sub-area, wet sediments, and due to the cohesive nature of these wet sediments, they stick here and drops them. And here are the governing equations. It's too complicated. If you want to know, I will tell you what it is. And the second approach, which is similar, but here we use a non-Newtonian fluid to represent substrate. It helps you save the computational costs because there are no numerous particles here. It is more efficient, and the organism is reproduced by multivariate discrete method, which will be a method. If you want to know more, I can tell you. So with this kind of approaches, we can reproduce the trace formation process on different environments and behaviors. As you can see here, this is just animations. This is a monopolic flora. This is a lady, the trace it creates, and this is the background fluid motion. This is a relief. The color is a relief of the trace. And we can do these simulations on different environments. For example, this is the trace created underwater. So these particles are not cohesive. They are just discrete particles on the water. And the trace morphology is quite uniform, quite straight, and quite smooth. And yeah, the surface is uniform. But if it's created on, for example, microbial EPS-infused sediments, then this surface becomes blurred. But if it is left on the subaerial wet sediments, you see the levee becomes slender, and there are some collapsing leaves. Yeah, this is because for wet sediments, there is a liquid bridge between two sediment particles, and the surface tension between the surface tension of this liquid membrane force two particles together. This makes the wet sediments very cohesive. If you use your hand, especially wetted hand into the sand, dry sand, the sand will stick to your hand. This is because there is tiny little bit of bridges between the sand and your hand, or between sands. So these features can also be observed in either fossil records or experiments. So this helps you to diagnostic the putative environment, and this kind of diagnostic can be independent to some ontological features. And it is also similar with deep forests, for example, if it's produced wet subaerial sediments, you can see this sharp, even concave structures. If it's produced underwater, it's a V-shaped section. And for just using that, the trackways, if it's produced aerially, there are some splashes. These splashes are very typical of subaerial environments. We can also use these numerical simulations to calculate, for example, the movement speed of the organism, because we find that the variance of the leaves increase basically linearly with the movement speed of the organism. So by measuring these the variance of the leaves, we can calculate the movement speed of the putative trace maker like this. We can use numerical simulations to do analysis. So let's reach to the conclusion now. In today's talk, I tell you something about the trace fossils, trace fossils and their paleontological significances. Then I introduce to you the frequency analysis and the coloration analysis of trace fossils, which could tell you the putative affinity of the trace makers, or at least its anatomy or its zoological tendencies. And we can also use these two metrics to quantify the classification of the trace fossils by making a 2D phase. Then I talked about something about the smoothness of the trajectory smoothness of the locomotory trace fossils and how it informs of the durability, the body size of the body plan, whether it's the sex or the sector range of the transporter, and see how this changes through the Cambrian transition. And lastly, I briefly talked about the numerical simulations that can be applied to the study of trace fossils, from pure mathematical simulations to those simulations that can reproduce the formation process of trace fossils. And this can help us, for example, calculate the movie speed or the diagnostic to find out the putative environments of the trace maker, like this. And it can also generate data for these analyses. And lastly, what I want to do is to build up a link between the biological signals of the trace maker, for example, separate, the body size, the mode, and the morphology of the traces, which is characterized by different metrics like frequencies, the coloration, the smoothness, the roughness, or the fractal index, like this. To build to link the biological signals with the morphology of the traces, with dimensional analysis. This is a very powerful tool that I don't have time to introduce one-dimensional analysis here, but it's much more powerful than artificial intelligence. So this is my final end of my work. Okay, this is all of my talk. Thank you, everyone. So if you have any questions.
Part of it at first, so this is my second time. Now I'm beginning to follow. Do you have any questions?
First one is we have a discrete definition about the behavior because many times we have the promotion with race. That you can the total of the promotion while including the amount of the promotion and the part behavior together, or do I consider second, if you know the paper of Wang and others, sometimes looking at some trace fossils are related to the amount of the cost, the organic matter. They use examples in the tailor parts and show that the amount of organic matter control in some way the morphology. This is one of the things that I find quite more difficult to follow the idea. Because you are establishing that animals is involved the animal or the behavior or the morphology that control all the things. How you control the amount of food that they need.
You mean
present day environment.
You mean yes, for example, it it's not a pure puzzle. It's relations. Organism.
Well, I have another question. question. I was wondering if maybe you or maybe Luca can tell us what are you working together or what are your future research projects? I just want to know about the collaboration between you guys.
We will establish. For example, Carol? Caroline.
Yeah.
With some technical. And you already have some sprites or Spartan structures. And you can see these Spartan structures tells you some information about the structure that produces the structure, whether it is rigid, whether it is soft. We examine the fossils. Soft. And whether they can produce forces. We discussed about that. There are some signatures that tells you these are passive. They can't control that structure. That structure lags behind the organism. So yeah, we can do some quantification of the morphology and just see. Even we can calculate the mechanical properties of that structure that produces sprites from this, and we can there is a phase difference between the uppermost morphology of that sprite and the basal burrow. So this tells you something about slapping. I forgot that word. Yeah, yes.
Okay. So any other questions? I think we present you with the certificate. Okay, thank you so much. Bueno, esperamos para el próximo ciclo de zonas ideas, que será próximamente, les avisamos por ahí. Muchas gracias por venir. Suscríbanse al canal. Suscríbanse. Stop. Sí.